Hello everyone and welcome. In this video we're checking out variant 1 of May-June 2023. Before starting, make sure to check out the free notes I have available for you in the comments section down below. Now let's see question number 1. The electron micrograph shows an onion root cells prepared using freeze fracture technique. The cells were quickly frozen and physically broken apart. Freeze fracture breaks apart cells along weak areas such as membranes and surfaces of organelles. So there's structure X, and which statement best explains the appearance of the electron micrograph? Okay, so what explains the appearance of the electron micrograph? So again, how freeze fracture works is that these cells are physically broken apart and it breaks apart the cells along the weak areas, specifically across the membranes. Okay, so it is definitely the technique that scientists actually use in order to separate the phospholipid bilayers. So what could structure X be? The cells were broken apart at the endoplasmic reticulum. Structure X is the ribosome. The cells were broken down at the nuclear envelope, and structure X is a nuclear pore. Or the cells were broken at nuclear envelope and the structure X is a ribosome. Or the cells were broken at the tonoplast and structure X is a plasmodesmata. Interesting. So what I would see here is that structure X is basically this circular shape, and one of the options said it's a nuclear pore, it's like a hole, and another said it's a ribosome, although a ribosome would be much more spherical if I'm being honest, and it's supposed to be breaking at a weak area, so an endoplasmic reticulum or the nuclear envelope. But what's the difference between the endoplasmic reticulum and the nuclear envelope? So there's no difference. It's not A or C. This just leaves me with the tonoplast or with the nuclear envelope. Now here he said this is a root cell, so yes, there's a chance that there is a plasmodesmata. Okay, but the cells broken apart were broken at the nuclear envelope, so structure X is the nuclear pore. Well, there are many pores, that's for sure. Okay, and so for us to assume that this is a nuclear envelope and that they're broken along the nuclear envelope and these are the nuclear pores, I suppose we can work with that, given that, plasmodesmata, they're extensions of the cytoplasm. It's really hard to break them. So I would have to say it's B. Which cell structures can form vesicles? Cell surface membrane, endoplasmic reticulum, Golgi body. So what can form vesicles? The cell surface membrane can by endocytosis. The endoplasmic reticulum can when it packages proteins, when it's releasing substances. The Golgi body can form vesicles when it's again releasing the substances it's just produced. So all of them can. Okay. 4 students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by number. Synthesis of polypeptides, so this has to be ribosomes. Okay, synthesis of lipids, the smooth endoplasmic reticulum. Packaging of hydrolytic enzymes that will remain in the cell. So I think packaging of hydrolytic enzymes, it could be the Golgi apparatus, I suppose, because they're enzymes, so they're functional. So let's assume it's Golgi. Okay. Let's see. Membranes which surround an enclosed cavity. This could work for the endoplasmic reticulum. Non-membrane bound spherical structures. This is definitely our ribosome. Double membrane interspersed with pores. Double membrane with a lot of pores. It's the nucleus, but it's definitely not here. Okay, non-membrane bound cylindrical structures. Non-membrane bound and cylindrical. I mean, this could be the centrioles, but okay. Membrane bound sacs arranged as flattened stack. Membrane-bound sacs arranged as flattened stacks. So let's see our options here. So it has to be between V, W, and Z. What's it called? Here he says 1 is either W or Z. So non-membrane-bound circle structures or membrane-bound sacs. It's definitely not membrane-bound sacs. The ribosomes are W, so it has to be between A and B. 2, V or Z. Membranes which surround an inner closed cavity. This could work for the smooth endoplasmic reticulum, yes. So yes, definitely we could choose this guy. What about the membrane-bound sacs arranged as flattened stack? I think this works for the Golgi apparatus. Membrane-bound sacs arranged as flattened stacks. So yes, I would say that 3 is Z, and so that's A. Which cells contain a tonoplast? Now, tonoplast is the membrane that surrounds the vacuole, the large permanent vacuole. Root hair. The root hair definitely has a large vacuole, so yes. Okay, companion cell. The companion cell also has a vacuole. The sieve tube element does not have a vacuole because remember, sieve tube element is hollowed out in the center with a thin cytoplasm towards the edge, no space for a vacuole. The endodermis, sure, the endodermis does have, okay, so it's going to be check, X, check. D. Which organelles found in animal or plant cells are surrounded by double membranes? Okay, double membranes, chloroplasts, mitochondria, vacuoles, chloroplasts and mitochondria and nuclei. Okay, moving on. Some scientists think that the mitochondria evolved from bacteria that entered the cytoplasm of a different cell and were able to survive there. What features of mitochondria support this hypothesis? Well, of course, features include the 70S ribosomes, the circular DNA in the mitochondrial matrix. I think these are the two features that are in common between mitochondria and bacteria. So let's see. Folded internal membrane. Yes, despite the bacteria do have a little bit of mesosomes, which are basically these tiny folds in their membranes, but they aren't as large as the folded internal membrane. Plus, the mitochondria is double membraned, so the bacteria don't really have an internal and external membrane, it's just a single membrane. So one is X. Okay, and circular DNA, yes. 70S ribosomes, yes. So D. What is present in all viruses, all prokaryotes, and all eukaryotes? All of them. Do they all contain ribose, or do they all contain deoxyribose, or cytosine, or thymine? Well, all viruses, all prokaryotes and all eukaryotes. Remember, there are two kinds of viruses. There's retroviruses, which only have RNA for their genetic material. There's also adenoviruses, which only have DNA for their genetic material. Now, this means that we can't just choose these two, okay, ribose or deoxyribose. Cytosine and thymine, remember the retroviruses that only have RNA, they don't have thymine, instead they have uracil. So we can also eliminate D, leaving us with cytosine. The table shows some steps that can be made in carrying out the Benedict's test. Which combination of steps is required to carry out a semi-quantitative test on the reducing sugar solution? Standardized volume of Benedict's solution and volume of test solution. Boil with hydrochloric acid and then neutralize it. Okay, so he said here that which combination of steps is required on a semi-quantitative test on reducing sugar. We don't need to boil it with hydrochloric acid. We don't need to carry out hydrolysis because it's already a reducing sugar solution. So we just need to standardize volume. Okay, we just need the Benedict solution, and we need to do some boiling with the Benedict solution and compare the final color with numbered colored standards. Exactly. So check here and there. C. Okay. The diagram shows 3 examples of different bonds. So you have bond 1, bond 2, bond 3, and so it said which bonds hold the secondary structure of proteins together. Now, secondary structure is going to definitely be hydrogen bonds. So it's not bond 2 or bond 3. Bond 3 is a disulfide bond, and bond 2 here is a peptide bond. Bond 1 is the one between hydrogen and oxygen. It's the bond we're looking for, so it's 1 only. Insulin is a globular protein involved in cell signaling. It's transported in the blood plasma from the cells that synthesize it to its target cells. A molecule of insulin contains 6 sulfur-containing amino acids and has 2 polypeptide chains. Which statement about insulin are correct? An insulin molecule has quaternary structure. So, which statements about insulin are correct? Now, firstly, he said insulin is a globular protein and it's transported in the blood plasma, and the molecule of insulin contains 6 sulfur-containing amino acids and has 2 polypeptides. So it has quaternary structure, it has 6 sulfur-containing polypeptide amino acids, which means it most likely has disulfide bonds, and since it's globular, that means it also has tertiary structure. So an insulin molecule has a quaternary structure, yes. Insulin polypeptides are held together by 6 disulfide bonds. Not necessarily 6, they contain 6 sulfur-containing amino acids and 2 polypeptides. So in total we have 6 sulfur-containing. So in general, if all of them were to engage in disulfide bonding, this would mean that we would have a total of 3 bonds between the polypeptide chains, because each chain is going to have 3. However, this is not to say that all amino acids with sulfur can form disulfide bonds. We know cysteine can, but there's also methionine that has sulfur but does not form disulfide bonds. So 2 is wrong. Okay. Amino acids with the hydrophobic R groups would be found in the center of an insulin molecule. Exactly. Remember, insulin is a globular molecule. There are hydrophobic amino acids towards the center, pointing inwards, and then hydrophilic ones engaging with water. So 1 and 3, that's C. Which statements correctly explain why blood plasma can be maintained at a stable temperature? It has low specific heat capacity. it's able to be maintained at a specific stable temperature because actually it's a high specific heat capacity. Now, latent heat of vaporization would be relevant if there was evaporation, but there isn't any here. We're not looking at plasma and how it evaporates. It's definitely B. Okay, which region on the graph shows the activation energy of the enzyme catalyzed reaction? So we're looking specifically for the activation energy with the enzyme. So with the enzyme, it's B. Without the enzyme, it's C. It's the higher one because the enzyme works to lower the activation energy. The graph shows the effect of increasing substrate concentration on the rate of enzyme catalyzed reaction. We have rate of reaction, substrate concentration, and then the maximum rate of reaction. And line P represents the result when the enzyme is used at optimum temperature and optimum pH and optimum temperature without an inhibitor. So there's no other limiting factor. It's just basically that maximum rate of reaction with the inhibitor, I suppose. Yes, so the reaction change conditions are changed. So what descriptions of changes to the reaction conditions could result in line Q if all other conditions were kept the same? So I would suppose that since we end up reaching the Vmax, since we reach it, we just reach it a bit at a higher we just reach it after lower rates of reaction. I suppose that basically in the end this is a competitive inhibitor. Okay, we didn't use a non-competitive inhibitor, so I don't think that it's about temperature, because if it's about temperature then it would plateau somewhere else, like this for example, because there's another limiting factor. But it's not about temperature. There's a competitive inhibitor, something preventing the substrate from reacting with the enzyme, and that's the competitive inhibitor. So what description of the changes to the reaction conditions could result in line Q? An inhibitor that attaches to the site other than the active site. This is wrong because it has to attach to the active site. That's the only difference between a competitive and non-competitive inhibitor, is that the competitive inhibitor directly attaches to the active site given it has a similar structure to the substrate. So this is the substrate, this is the competitive inhibitor, and the enzyme couldn't tell the difference. So this is why the rate goes slower, because the enzyme is wasting time with the competitive inhibitor rather than reacting with the substrate. Okay, so this is wrong. An inhibitor that has a similar shape to the substrate, yes. An inhibitor that blocks the active site of the enzyme, yes. Carry out the reaction at higher temperature, no. So it's 2 and 3 only, that's D. Which statements about cell signaling is correct? One type of receptor molecule recognize all ligands in the body. A single receptor complementary to all the ligands. Definitely not. A receptor is specific to a single ligand or a single signaling molecule. The binding of a ligand may cause a change to the shape of the receptor. A change to the shape of the receptor, I suppose, well, it doesn't really cause a change in the shape of the receptor. We're gonna get back to this. The receptor of ligands are always found on the inside of the cells. Actually, somewhere on the inside, like the receptors for the ligands that are made of that are specifically maybe hormones, the ones that like that. But otherwise, it's gonna be also on the outside. The same ligand is made by all cells in the body. No. Okay, the same ligand is not made by all the cells in the body. Some cells produce specific ligands and so on. So yes, the binding of the ligand may cause the change in shape of the receptor. A conformational change in shape is normal whenever something binds to another. So B is the answer. 4 students A, B, C, D observed plant epidermal cells that had been placed in a concentrated sucrose solution for 30 minutes. They were asked to identify the partial permeable layer, and explain the appearance of the cells in terms of water potential and the movement of the water. So firstly, what is the partially permeable layer? It's definitely the cell surface membrane, not the cell wall. The cell wall is fully permeable to water. Okay, water potential at the start of the experiment. So they say here cell contents have a lower water potential than the sucrose solution, and then movement of water during the experiment. So remember, they are concentrated sucrose solution. So actually the contents should have a higher water potential. But let's see what it says here. So have a lower water potential, and water moved out of the cell and no water moved in. Okay, so we have a lower water potential. Why would water move out of the cell? Remember, in such scenarios, I would like to draw. Okay, so this is, let's say, the plant cell. Okay, now the plant cell here, he said that we've placed it in a highly concentrated solution. So the water potential here is low, the water potential here is high. And water likes to move according to osmosis from high water potential to low water potential. So like this. Okay, but here he's saying that it's the opposite, and instead of water moving in, water is still moving out. So this is wrong. Okay, moving on to B. Cell contents have a higher water potential than sucrose solution. Yes, water moved out of the cell and then moved in. Okay, that's more water moved out than moved in. Fantastic. Moving on to 16, the table compares the surface area to volume ratios of 5 agar blocks that differ in dimensions but which all have the same volume. The agar blocks can be used to measure the efficiency of diffusion, where efficiency is measured as the time taken for a dye to reach all parts of the block, the length, the width, the height, the surface area, the volume, and the surface area to volume ratio. And he said here, which prediction can be made about the way in which size and dimensions of these blocks affects the efficiency of diffusion? Well, what's it called prediction that we can make? Firstly, you could see how when the surface area, okay, so we have different surface area to volume ratios, and what we know is that, okay, they do not really provide us with anything, but they just, okay, what do you predict as a person? Well, I know that when surface area to volume ratio increases, okay, what happens is that diffusion rate increases, and when when you make the block size smaller, like here you can see the width is 4 and 2, okay, you had a greater surface area to volume ratio, like here for example 4.1 when we had the smallest height. Okay, and so yes, the surface area to volume ratio increases when you decrease the overall size, and the diffusion increases when the surface area to volume ratio increases. So the efficiency of diffusion will decrease as the width of the block increases. So you can see how, for example, with like a higher width, it was still quite a high rate of diffusion, and based on this information, a prediction like this would not be accurate, because whenever you saw, for example, an increase, it was always an increase in the rate of diffusion. So we can see the other options. The efficiency of diffusion will increase as the height of the block increases. Now you can see that when we had the lowest height, we had the highest surface area to volume ratio. So based on these results, again, I don't think we can make this prediction. The efficiency of diffusion will increase as the block of fixed volume is flattened. Okay, flattened. So if you're flattened, now he gave you the volume is the same, it's just that when the surface area is bigger, which means it's flattened, this means that the surface area to volume ratio is bigger, and so yes, we can make this prediction. The efficiency of diffusion will decrease if it's elongated. I feel like elongated and flattened are synonyms in this point. Like when you elongate someone, when you stretch it out, you're basically flattening it, so this should increase the diffusion, but here he's saying the opposite, so C is my answer. The cell cycle includes mitosis. What are the features of this type of nuclear division? Form cells of equal size to the parent cell. Actually, no, they don't form cells of equal size to the parent cell. They form cells that are genetically identical to the parent cell, and right when the cells are formed after, they're going to actually be a whole lot smaller than the parent cell. Then they're going to grow in the first growth period after mitosis. So no. Forms genetically identical nuclei, yes. Semi-conservative replication of DNA. Well, replication of DNA is definitely part of the cell cycle. However, since he's asking about nuclear division, about specifically mitosis, this is only part of interphase and not part of mitosis or nuclear division, and therefore it's 2. A student observed the cells in the growing region, the meristem of an onion root, and obtained data shown. So he got the different stages, and he said which percentage of cells contain chromosomes that appear as 2 chromatids. Remember, this is a repeated question, so make sure to memorize how to solve it. Firstly, we need to understand that to have 2 chromatids means to have passed the S phase, to have basically done DNA replication. So that's going to be during interphase, but towards more of the end, because actually 90% of interphase is G1, is the first cross period. So if I can't really count the cells in interphase as cells with 2 chromatids, because 90% of them are actually going to have 1 chromatid, so we don't count these. We count the cells in prophase because it's going to happen after the interphase and also in metaphase. However, we don't count anaphase or telophase, because right in anaphase the chromatids are going to split, so they're going to no longer be together, and so we only count like this: 73 plus 16 divided by 886 plus 73 plus 16 plus 14 plus 11 times 100. So basically we divide this number of cells by the total number of cells. So 73 plus 16 divided by 886 plus 73 plus 16 plus 14 plus 11 times 100 is actually 8.9%. B. Which statement about messenger RNA is correct? In eukaryotic cells, mRNA is made by removing exons from the primary RNA transcript. That is actually right, okay? During RNA splicing, mRNA is going to basically be edited in order to become more, efficiently, it basically become more practical so that the cells can encode it directly, translate it directly, so this is true. mRNA is a single-stranded polynucleotide containing a different purine base than DNA. Actually, it's a pyrimidine base. What's it called? mRNA contains uracil, whereas DNA contains thymine. Both thymine and uracil are pyrimidines. Okay, mRNA molecules contain ribose sugars joined to phosphate groups by phosphodiester bonds. So contain ribose sugars joined to phosphate by phosphodiester bonds. That is actually true. Okay, what's it called? Oh, okay, I did not notice he said removing exons. So yes, I was confused as to why both statements were right. They were not. The first statement says removing exons, which, okay, the pencil is no longer, okay, yes. Exons are the coding parts of mRNA. They're not the ones removed. What's removed is actually the introns from the primary RNA transcript. So this is wrong. This is also wrong. And lastly, the monomers of mRNA contain a phosphate group, a deoxyribose sugar, that's wrong, and nitrogenous base. Okay, they contain a ribose sugar. Which structures are involved in transcription only? DNA template strand, anticodons, RNA polymerase. Okay, structures can, involved in transcription? Yes, DNA template strand. Anticodons are involved in translation. RNA polymerase is definitely the thing that basically carries out the transcription. So it's the enzyme that carries out transcription. 21. One gene provides the code for the production of what type of molecule? Well, a polypeptide, of course. Okay. The table shows some mRNA codons that code for certain amino acids. He said the DNA template strand has the base sequence shown: ACA, GTA, TTA, and so on and so forth. He said what would the change in the amino acid be if the first base in the fifth DNA triplet was substituted for an A base? Interesting. So let's see. Let's separate the triplets, and he said that the fifth one, 1, 2, 3, 4, 5, this one over here, if we substitute the first base, the G, for an A, so instead of GCA, we have an ACA. So let's see. ACA, now here this is the mRNA codon. Okay, we're looking for DNA because this is a DNA template strand. DNA is complementary to RNA. So GCA used to be CGU. Yes, and CGU is which one over here? It's arginine. Okay, so we had arginine. Now we have ACA, which is basically UGU. Now UGU is cysteine. So instead of arginine, we have cysteine. So arginine, cysteine. Which structures contain cytoplasm with mitochondria and the nucleus? Okay, the xylem vessel, the companion cell, the phloem sieve tube element. A nucleus, a mitochondria, and cytoplasm, it's only A, because obviously this guy over here does not have, it does have, cytoplasm, and it does have, it does have mitochondria. However, it's, the xylem vessel element does not have any of these features. It's a dead part. The phloem sieve tube element does not have a nucleus, so it's A. What is the correct term to describe intermolecular hydrogen bonding between water molecules? Adhesion, cohesion. So between water molecules, that's going to be cohesion. Between water molecules and a surface, that's adhesion. Water movement from region of high water potential to lower water potential across the partially permeable membrane, that's osmosis. Diffusion, I think you all know the definition of diffusion. Our famous question. The diagram shows the outline of a xerophytic leaf that has been left for 45 minutes in different conditions, P and Q. So remember, guys, this is a very repeated question, and I need you all to have memorized this, okay? Which statements about the cells in layer Y of the leaf in each conditions, P and Q, after 45 minutes are correct? So there is a less negative water potential in P than in Q. Okay, remember, here the cells have absorbed lots of water, so they were placed somewhere that has a high water potential. So yes, overall they're going to have a less negative water potential because, as you can see, it's all flat because it's absorbed so much water, whereas here it's shrunk up because it lost so much water, so the water potential here is actually lower. So this means that it's more negative, whereas here the water potential is higher. Okay, the cells may be turgid in P and plasmolysed in Q. Yes, the cells here have been placed in a dilute solution, they're going to be turgid. Cells here in a concentrated solution would be plasmolysed. Cells are less turgid in P than in Q. No, cells are definitely more turgid in P than in Q. There is no net diffusion of water into Y. in what's it called, in either P or Q. Now this is, I think, the confusing point for many students. Yes, after 45 minutes we will have reached equilibrium. All the possible water would have left here, and so now the water potential inside is equal to the water potential outside. All the possible water could have entered here, and so water potential outside equals to water potential inside. So 1, 2, and 4, A. How does sucrose move from chloroplast to the phloem? Well, from the chloroplast to the phloem, firstly it's going to be produced and everything, and then it's going to leave the cell, and enters the companion cell via co-transport or indirect active transport. And then it's going to move through the cytoplasm of the companion cell and into the sieve tube element through the plasmodesmata. Normal diffusion here. So let's see. How does sucrose move? Diffusion, yes. Aplast, sure. Why not? Of course, it could move through the cell wall if it likes to. And the Symplast pathway, yes. So it's 1, 2, and 3. Companion cells involved in loading sucrose into the phloem sieve tube elements. Okay. Companion cells involved in loading sucrose into the phloem sieve tube elements. Now, actively through the co-transporter proteins and passively through the plasmodesmata. Yes, actually, they are going to actively, by indirect active transport, enter the sucrose with the hydrogen ions through co-transporter proteins, and then the sucrose moves across the cytoplasm and through the plasmodesmata and into the into the sieve tube elements. Okay, actively through the co-transporter proteins and plasmodesmata, definitely not actively through the plasmodesmata, passively through the co-transporter proteins only. Okay, I think A is the answer here. The photomicrograph shows blood cells as seen using a high-power light microscope. He's labeled 1 and it's a phagocyte, and 2 and let's see what we have here to label. So which row identifies the type of white blood cell shown? So that 1 is definitely a neutrophil, and I think 2 over here is very huge to be a monocyte. I think 2 has to be a a macrophage. So let's see what options we have. 2, either monocyte or neutrophil or monocyte or lymphocyte, and then 3 is either neutrophil, macrophage, lymphocyte, or monocyte. Okay, so I think that this here we need to actually go back to the book and review if we have a hard time identifying the cells. But yes, 1 is a neutrophil, 2 is a monocyte, and 3 is a lymphocyte. So C is our answer. Moving on to, and remember, what are monocytes? Monocytes are the cells right before macrophages. So yes, my understanding that this is a macrophage is correct, and actually macrophages are slightly larger. Okay, moving on to plan diagrams of 2 blood cell vessels are shown. So fatty tissue and everything. Which labels are correct? 1, aorta. Okay, so let's see what are the labelings here. So he's asking us 1 versus 2. So aorta, artery, vein, coronary artery. 2, vena cava, vein, artery, coronary vein. And then 3, elastic fibers. So let's see. So 3 is the outer layer. And so in the outer layer we should have collagen in both. Okay, so yes, collagen. And then in the middle layer, I think that's 4, smooth muscle and elastic tissue. And in the inner layer, it's endothelial cells. Okay, so endothelium, smooth muscle, and collagen. And then he's asking us what is 1 and what is 2. Well, it's definitely not vein and artery. It is artery and vein. And is it aorta and vena cava? Well, the aorta is a whole lot larger than the vena cava, so I don't think so. And coronary artery and coronary vein? Well, there isn't really a way for us to tell right now the coronary artery or coronary vein. But, I feel like the layers aren't as thick as they should be. Let's go back to this later. So it's definitely not aorta and vena cava. It's artery, vein, and even if it's the coronary artery and coronary vein, these things, they're labeled wrong. Okay, what are found in blood and tissue fluid? Carbon dioxide, fatty acids, white blood cells, proteins. In blood and tissue fluid, yes, there is carbon dioxide. Fatty acids, are they found in blood and tissue fluid? Definitely. Fatty acids could be found in blood and tissue fluid. White blood cells are, yes, they both are found in this and that. Proteins, yes, actually, even though it might sound confusing. How do you mean there's proteins in tissue fluid? There are proteins in tissue fluid, but does not mean that these proteins have left from the blood, and it also doesn't mean that they are big proteins like the the globular proteins, globulins and everything. So 1, 2, 3, 4. Which reactions will be slowed down by an inhibitor of carbonic anhydrase? Carbonic anhydrase is an enzyme that catalyzes the formation of carbonic acid from CO2 and H2O. So this reaction over here should definitely be affected. However, there's no option for one only, so we need to choose another one. While CO2 and hemoglobin to carbonic hemoglobin has nothing to do really with carbonic anhydrase. However, H2CO3 turning into hydrogen ions plus carbonate ions, you can see basically here that H2CO3 is the reactant. So I suppose that since this is the product, and if we inhibit this reaction, there will be no more carbonic acid. This means that this guy will also be affected, I suppose. So this would also be slowed down. So it's 2 and 3, which is D. Which sequence of letters correctly identifies the order of events during the cardiac cycle? Atrial walls contract, impulse. Okay, so first we need the sinoatrial node. So wave of excitation spreads from sinoatrial node, X. Okay, this is followed by wave of excitation enters the atrioventricular node, or wave of impulse is delayed by a fraction of a second, or what's it called? Ventricles contract. Okay, what happens next is that the atria are going to contract, so that's going to be T, and then this is going to be followed by the wave of excitation enters the atrioventricular nodes, and then the wave is going to be delayed. After that, it's going to enter the Purkinje tissue and the ventricles are going to contract. So let's see, X then Y, that's wrong, X then U, X then T, then V, then U, then W, then Y, C. Okay, or X and V, okay, fantastic. The diagram gives information about blood pressure in left side of the heart during the cardiac cycle. So you can see how the pressure is going to increase from these points on, and there are different labels here. So you have aorta, atrium, ventricle, and valves open and close at points numbered, which will identify as valves opening or closing at the points numbered. Okay, so since this is the ventricle, this means that when the pressure is going to be super high, this is because the pressure is increasing here, that we have to understand this is ventricular systole. Now, ventricular systole, they also labeled other things. There's the aorta, which is right here, and the pressure increases all of a sudden from 0.2 to 3 in the aorta, which means this is when the blood enters the aorta. So the ventricle starts contracting at 1. Okay, so I will assume the atrioventricular, the atrioventricular valves close here. And then here the semilunar valves open because this is when blood enters the artery. And then here they close, and then here the atrioventricular valves open again because we have reached generalized diastole. So which row identifies the valves opening or closing at the points numbered? So atrioventricular valve opens at point 4. Semilunar valve opens at point 2, atrioventricular valves close at point 1, and the semilunar valves close at point 3. Exactly, so D. A person with no breathing conditions rests for an hour, and the breathing time is shallow and slow, so little air from outside reaches the alveoli, and the person's heart remains constant. What statement is correct? The carbon dioxide concentration in the pulmonary vein will be higher than the pulmonary artery. This is wrong. The pulmonary vein is oxygenated, which means it has lower CO2 compared to the pulmonary artery. CO2 molecules in the air of the alveoli move out of the blood by active transport. Absolutely not. It's normal diffusion. The air in the alveoli has a lower concentration of oxygen in the blood than in the pulmonary vein. Actually, no. The oxygen in the pulmonary vein comes from the alveoli, so it actually has a higher or equal oxygen concentration. Oxygen molecules diffuse from air in the alveoli into the blood at a slower rate than when the person is active. Yes, of course, when the person is active, they are producing more CO2, they require more oxygen, so the concentration gradient will be steeper for diffusion of oxygen and CO2. So, of course, D. Which statement about all bronchioles are correct? They have epithelium. Yes, they do. They have goblet cells. No, they don't have goblets or cilia. They have muscle tissue. Yes, smooth muscles, 1 and 3 only. Diagram shows some pathogens that cause disease in humans and some ways that are transmitted: bacterium, protococcus, pathogen, and virus. Okay, so here he says method of transmission: contaminated water, mosquito bites, coughing and sneezing. Okay, the protococcus will be Plasmodium, which basically causes malaria and transmits by a vector, which is a mosquito. So this is 2. Coughing and sneezing could be tuberculosis, bacterium, so that's 1. Contaminated water. Okay, actually contaminated water could be the bacterium instead, because It could be cholera, for example, so that's 1. Whereas coughing and sneezing could be just basically a rhinovirus, and so it's 3. Okay, now he says here, what is the correct method of transmission of malaria? It's 2 and X. Okay. Some of the processes which result in the formation of population of bacteria that are resistant to a new antibiotic are listed. Which is the correct order of these processes? Change in reproductive success of bacteria, increase in frequency of the resistance alleles in the population, increase in genetic variation within the population, random mutation occurs in the bacterial DNA. Exactly. So there's a mutation, and there will be, of course, increase in genetic variation within the population. Okay, so a change in reproductive success of the bacteria, I would have to say it's going to be first, after the mutation happens, this increase in genetic variation. Then change in reproductive success is going to be something that happens, so there's more genetic variation because the mutation happens. After that, not all bacteria are going to survive, only the ones that have this mutation, and then obviously this will increase the frequency of the recessive allele. So D. This moves me on to 38. When bacterium are grown in a petri dish containing a disc with antibiotic, there will be zones of the inhibition of bacterial growth. So you have antibiotic disc in zone of inhibition, area of bacterial growth, and the chart shows the sizes of the zones inhibition when a species of bacteria was incubated on 5 different plates of agar, each containing a disc with different antibiotics. So we have antibiotics and zone of inhibition, and you can see how if this is the area of bacterial growth. Now, the antibiotic stops the bacteria from going, and this is the zone of inhibition. The bigger the zone of inhibition, the less resistant the bacteria is. The smaller it is, the more resistant. And so the least effective antibiotic should be number 2, whereas the most effective should be the one with the biggest zone, which is number 3. So 3 is the most effective and 2 is the least effective. Now, which blood cell does not recognize, engulf, and digest non-self particles? So macrophages do that, neutrophils do that, phagocytes do that. T killer cells actually don't. Instead, they're going to basically bind to the receptors, or bind to the antigens, on the pathogen, and then basically insert insert substances that are going to kill and harm the pathogen. So this includes maybe hydrogen peroxide, just any type of material of that sort. So it does not really do the engulfing and digesting, does not do endocytosis. Now, repeated infections with malaria result in more effective immunity to malaria. Which type of immunity is responsible for effective immunity? So since it's repeated infections, this means the person gets malaria, develops an immune response, and then gets it again and has memory cells and everything. So since memory cells are produced, it's definitely active, and since they get it naturally, then it's naturally active. So that's C. Okay, and this is it for this video. I hope this paper was useful for you guys. If it was, make sure to like, share, and subscribe, and I hope to see you guys in my next one.